to show you so you can start thinking about it, because you have to make a 1-minute video with this, all this information in it. What's next? Interface. What else? Good, good. So that part sounds like everybody's got. What I do want to spend some time with is to make sure that you all can describe each one without reading out of a book. So tomorrow when you come in here, reading out of the book is not going to be fast enough. Okay? And we're going to see your hands doing something. So you don't have, how do I put this? You're not going to be able to read. You're just going to have to know what's going on. So it's going to look something like this. You're going to start off with, it's my cell membrane, and these are my chromatids inside of my nucleus. This is an animal cell, so I've got centrioles. This cell is an interphase, so I'm going to start and check. I'm in interphase, and I'm in the G1 phase of interphase, which is for growth. You were going. In the G1 phase of interphase, cell will grow and add organelles, lysosomes, and a rough endoplasmic reticulum, maybe a smooth Golgi apparatus. So basically the cell's just living, maintaining life. Maybe some ribosomes. So I'm in the G1 phase. Now let's say the cell next door in this animal died, and now I need to replace that animal cell that was there. So I'm going to enter into the S phase, which is for DNA replication, telling your people what you're talking about. And I'm going to make sure that I copy all of the sister chromatids identical, so I get all the genetic information so I can pass them on to the cell. Okay, so during S phase, all the DNA is copied, and I've got all the DNA I need to now enter into the next couple phases. Okay, so copied all the DNA in the S phase. In the G2 phase, basic premise is I'm going to start saving up, and you'll see in your game today, I'm going to start saving up a bunch of ATP and also start storing up some, or gathering a whole bunch of glucose so I can make more ATP. Okay, so I'm going to need a lot of energy, and that energy comes from the G2 phase, preparing for mitosis. Mitosis is prophase, metaphase, anaphase, telophase. So my next phase that I'm going to enter is prophase. So I'm going to move into prophase, and the nuclear envelope starts to break apart and disappear. Okay. Eventually I start seeing the chromosomes. Centrioles will migrate or move to the opposite ends of the cell. Notice the organelles are really not doing anything. But now I see chromosomes. Spindle fibers will start to appear. Okay. And I'm drawing these in a certain way on purpose. I'll see these little spindle fibers forming. And eventually the spindle fibers will pull tight with the centrioles, and I'll get all my chromosomes, and this apparently has 3 chromosomes, on the metaphase plate. Okay, so now I am in metaphase. I know it's metaphase because all the chromosomes are lined up in the center. The spindle fibers are hooked up to their centromeres, and the spindle fibers are going to pull those centromeres apart and sister chromatids apart in a phase called anaphase of mitosis. So here's what's going to happen, and if you do this just right, it looks amazing. The centrioles will pull the spindle fibers and the sister chromatids, this one's going to look awesome, apart to the opposite ends of the cell. Okay, so now anaphase is over because movement stopped. So I'm now in telophase. In telophase, I start reforming a nuclear envelope for each set of sister chromatids, and eventually 2 new nuclei completely reform. Okay, and my chromosomes start to break back down into regular old DNA, and I get a nucleolus reformed. There's my nucleolus. While that's going on, the cell membrane starts to pinch apart. Okay, and this is cytokinesis. So as we're pulling these apart, and the spindle fibers broke down in telophase, we finally get to the part where cytokinesis breaks each cell apart, and I have 2 separate cell membranes. And that's the end of the cell division phase. So you might say something like, this is the M phase or the cell division phase, cytokinesis plus mitosis. And then both cells now are starting over in interphase. deposit.